For most of the students in Sri Lanka, biology is a challenging subject because most of the students are not learning the underlying concept behind the theory in your resource book. Students remember to study the concept and be conceptual than be by-hearted. So, we will start the 2021 MCQ analysis, the question 1 with the underlying concepts. Question number 1, 2021 MCQs, the part 1 biology. Which of the following is a non-reducing sugar? The answers go as ribose, answer 2 lactose, answer 3 maltose, the fourth answer galactose, and fifth answer sucrose. Now, students, can you think the underlying concept that the question is based on? Actually, it is a laboratory practical based question. We will try to understand the concept behind the question. Remember, sugars are divided into 2 groups, reducing and non-reducing. This classification of sugars is based on the reducing and non-reducing nature of the sugars. Now, how do you detect the sugars reducing or non-reducing nature? We will understand. I told you that it is laboratory practical based. So, the basis is Benedict's test. That you use to identify reducing sugars. Students, how do you perform the Benedict's test? You add Benedict's reagent to the solution that you want to test for reducing sugars. So, you are going to find the presence of the reducing sugar in this medium and you are adding the Benedict's reagent into it. You usually add 1:1, the volume ratio. So, if you add 1 centimeter cube of Benedict's, 1 centimeter cube of reducing sugar solution should be taken. After adding Benedict's to the solution to be tested for reducing sugars, you should mix well and then boil it in a boiling water bath. Initially, the color would be blue because Benedict's solution has cupric ions. You know cupric ions in the aqueous condition, blue in color. So, gradually the color change will take place from blue to brick red if the reducing sugars are present. We will try to understand the concept behind this. Students, Benedict's reagent has copper 2+ ion or the cupric ion. When the reaction is carried out, the cupric ion is getting reduced to cuprous ion. As you can see, the oxidation number has dropped. So, this is a reduction reaction and this reduction is carried out by the sugar that you had added. So, during this process the sugar is getting oxidized. As sugar had helped reducing copper 2+ ion to copper 1+ or cupric to cuprous state, thus sugar is getting oxidized as this is a redox reaction where one reactant is reduced and the other reactant is oxidized. Here sugar had helped the reduction of the cupric ion to cuprous ion. So, the sugar is classified as the reducing one. So, it is a reducing sugar. So, now we will understand the list of reducing sugars in your syllabus. All the monosaccharides and the disaccharides except sucrose are reducing in nature. So, all the monosaccharides, the glucose, galactose and fructose, and the disaccharides except sucrose, the maltose and lactose. Are reducing. Hope you understood the concept behind the Benedict's test, students. Thus, how you classify sugars into reducing and non-reducing ones. So, the only non-reducing sugar you know is therefore sucrose. So, your answer goes as the answer number 5, the sucrose. Hope you understood the concept behind the question number 1. At the same time, without going into question number 2, I thought of explaining you the concepts behind the other 3 major tests identifying biochemicals. We will understand the concept behind the starch test. So, what is the name of the test for identifying starch, students? It is the iodine test. We will understand the principle behind that as well. So, you get a piece of food that you are going to check whether the starch is present. Here I took a piece of potato and I am adding few drops of iodine reagent. What will happen if the starch is present? It will go blue-black. So, blue-black coloration would be developed if starch is present. Now, what is the underlying principle or concept behind this test, the iodine test for starch? We will understand. Students, you know starch is a mixture of 2 polymer molecules, the amylose and amylopectin. So, there are 2 forms of polymers added together to form the starch. So, it is a mixture of 2 different molecules which are polysaccharides. Amylose is a linear molecule, whereas amylopectin is a branch molecule. This linear molecule is unbranched and it is helically coiled. So, it is a linear molecule, unbranched and helically coiled. And when it comes to amylopectin, it is a branch and that is not needed for the test result to be shown. So, the iodine test is completely relies on the presence of amylose in starch. We will see how. Students, iodine molecules in the iodine reagent will now stuck in the core of this helix like this, produce in a complex which is blue-black in coloration. So, when you add iodine reagent, if you get blue-black coloration, that proves the presence of amylose and then in turn the presence of starch. I hope you understood the concept behind the iodine test as well now. Right, moving to the third test for proteins, we will understand the concept behind that too, because when you learn with the concepts, it will go to your long term memory and it would be very much easier to memorize when the exams are at hand. Right, biuret test for proteins. Right. So, what is a protein, students? Protein is a polymer, biopolymer, where you find amino acids being peptide linked. So, your biuret test relies on the presence of the peptide bonds. If I give the formula of the peptide bond, it is CONH, and the biuret reagent again contains cupric ions in alkali, and the cupric ions will react with nitrogen, producing a violet complex. So, violet coloration is due to this complex formation between cupric ion and the nitrogen atom in the peptide linkage. So, that is the biuret test principle. Coming to the last, test for lipids. So, you know the lipid can be tested using the Sudan's test, Sudan 3 test. Or even Sudan 4 test. Remember Sudan dye, to produce a Sudan 3 reagent, Sudan, the red dye, is dissolved in ethanol. You know then the Sudan is a non-polar solution. Sudan solution is a non-polar solution. And you know lipid is also non-polar. There is no polarity in the lipid molecule. It is also non-polar molecule. The concept is like dissolves like, means polar solutes dissolved in polar solutions or polar solvents, and non-polar solutes dissolved in non-polar solvents or solutions. So, thinking about that concept, you can see here, when you add Sudan 3 solution to a lipid containing tube, if lipid is present, as Sudan 3 is also non-polar and the lipid is also non-polar. So, non-polar dissolves in non-polar, like dissolves like concept could be applied now, and Sudan 3 dye would color the lipid in red and you get red droplets being found. If lipid is absent, Sudan 3 would separate and would not color the solution. So, that is the basis of the Sudan test. I hope you understood all the 4 concepts behind all the 4 tests. The test for reducing sugars, test for starch, test for proteins, and the test for lipids. Thank you.